Morning. I'm sorry you're going to have to wait for the food. Well, we'll go ahead and get started. Today, it's a real pleasure to introduce a colleague and a friend, Nancy Braverman. She is actually a genetic counselor and did her genetic counseling training at Tulane, at Sarah Lawrence. And then she went to medical school at Tulane and did her pediatric residency at Yale and her chief residency in Baltimore. After that, she did her clinical and biochemical training at Hopkins and worked there for a while, and then we lost her to McGill. So today, she's an associate professor at McGill University and is here to share with us her, her experience in trying to detail that with the peroxisomal disorder. So it's with great pleasure I introduce Nancy Braverman today.
Okay, thank you. I'm sorry that you have to forego breakfast, but I do too, so. So I'm going to talk today about one of my favorite topics, peroxisome diseases. And I came down here, I'm very happy for this respite from this sub-zero temperatures in Montreal. And I am wondering, this is my second winter, and I'm wondering when I'm going to become as acclimated as the Montrealers, because nothing keeps them indoors, as you can see, and to my amazement, they're even biking in blizzards. So when Ronnie invited me to come down here, she was talking about this vision of this educational talk conferences, and I thought that that was a great idea. And her project was really to bring a set of educational lectures to a diverse group of people involved in genetics and to inspire. So I thought that this was a good thing to participate in, and came down here. So the learning objectives that I set out today was to review and get everybody up to date on peroxisome disorders, develop a diagnostic approach based on specific biochemical and genetic defects, and to appreciate challenges in developing therapeutic approaches for these patients. Now, as Ronnie said, I'm trained as a metabolic geneticist, so I see a diversity of patients with metabolic disorders, and then in the lab I work on bench work on a translational program in peroxisome diseases. So I'm going to talk to you sort of about the clinical perspective mixed in with the research and how we've contributed to understanding these disorders at the bench, and then sort of back to research in looking for therapy for these disorders, and then back to the bench. So this is a timeline of the work in this field. And peroxisomes were first described as microbodies by a Belgian biochemist who won a Nobel laureate for his work in peroxisome and lysosome and subcellular organelles. His name is Christian de Duve. And then in 1964, a neurologist from Iowa, Hans Zellweger, described a group of patients with similar findings in the brain, liver, and kidney. And so a malformation syndrome that was termed the Zellweger cerebrohepatorenal syndrome came into being. And it wasn't until about 10 years later that a pathologist from Albert Einstein in the Bronx actually noted that there were abnormal peroxisomes in tissues from patients with Zellweger syndrome. And then another decade later, when we learned about metabolic pathways in peroxisomes and that those were also defective in Zellweger syndrome, we were able to tie the etiology to Zellweger syndrome to defects in these metabolic pathways. And so Zellweger syndrome, or the peroxisome biogenesis disorders, became a paradigm for metabolic malformation syndrome. So in the late 80s, Hugo Moser at Johns Hopkins began collecting cell lines from these patients, and he complemented them by fusing the cells and determining if there was rescue of peroxisomal function. And if there was rescue, the idea was that the patients had defects in two different genes involved in peroxisome assembly, and if there was no rescue of metabolic defects, the idea was that the patients had the defects in the same gene. And in that way, he found out that there was a number of complementation groups in the peroxisome assembly disorders reflecting genetic heterogeneity. And in 1990, a number of investigators identified peroxisome biogenesis genes to yeast. And in the mid-90s, when the database of express sequence tags at NCBI was being put together, we used that to pull out human genes for peroxisomal disorders by probing with the yeast homolog. And we had these cell lines that Hugo Boser had banked, in which we could express the human candidate genes in these cell lines, see if they would recover function, and then look for the mutations in the cell line in that particular gene. So in that way, most of the human genes for peroxisome assembly disorders were identified. And then in this decade, we've really been studying protein functions, pathophysiology, and therapy. So these are just some diagrams of peroxisomes to show you what they look like. On your left is peroxisomes in controlled human fibroblasts, and these cells are fixed and permeabilized and stained with antibody either to the peroxisome membrane or to a peroxisome matrix protein inside, since they're permeabilized, and you get the same picture. And there are about 400 or 500 peroxisomes in normal human skin cells, and they're spherical, single membrane bound. They're present in all eukaryotes, and on the right is an EM from liver, and it shows you the size of the peroxisomes, which are the black structure in comparison to the mitochondria and to the glycogen granules. So there are a number of different metabolic pathways in peroxisomes, and probably more than 50 enzymes. And it may be that not all of the mammalian peroxisomal enzyme repertoire has been identified. And as we go through this list, just keep in mind that peroxisomes usually don't have the entire enzymatic set of the pathway, but only a portion of it. So these reactions go in and out of the peroxisome, and so there's there needs to be metabolic cooperation with other organelles and with pathways in the cytosol. So peroxisomes are involved with fatty acid oxidation, and that includes very long-chain fatty acids and branched-chain fatty acids like titanic acid. And they're also involved in the detoxification of hydrogen peroxide, which are produced in the first step of beta oxidation in the peroxisome by an oxidase, and that's how they get their name, peroxisomes. And they also have an enzyme catalase that breaks down the H2, the peroxide, into water. And peroxisomes are also involved in the synthesis of several compounds. One of these is docosahexaenoic acid, which we're all familiar with, I think, probably in this group. It's long-chain polyunsaturated fatty acid, and it's now being added to infant formulas as enfamil with lipo. And peroxisomes are also involved in the synthesis of bile acids and in the synthesis of a special class of membrane phospholipids called ether phospholipids, or plasmalogens. They are probably involved in the pre-linosterol pathway of cholesterol synthesis. And they're involved in the disposal of some amino acids like glyoxylate, which is derived from alanine and glycine, and also in the catabolism of glycine through glycolic acid. So this is just sort of a sampling. So these peroxisomal enzymes are also called peroxisomal matrix proteins, and they are directed to the peroxisome by virtue of having these cis-actin targeting signals. And most peroxisomal matrix proteins have this C-terminal SKL, or serine lysine leucine, or variant thereof. And the peroxisome receptor that recognizes these proteins in the cytosol is called PEX5. And a small number of peroxisomes have an N-terminal, bit more complicated signal that's probably more of a structural motif. And this pre-sequence is actually cleaved internally, and there are only 3 matrix proteins in this class: thiolase, which catalyzes the last reaction in beta oxidation, and I'll show you this, and phytonylchloride hydroxylates, which is involved in phytanic acid oxidation, and one of the enzymes involved, one of the 2 enzymes involved in plasmalogen synthesis in the peroxisome, HEPS, or alkyl glycerol phosphate synthase, and the receptor for these PTS2 proteins is PEX7. The other interesting thing about peroxisomal matrix proteins, in contrast to mitochondrial matrix proteins, is that they're imported as oligomers. enzymes. A huge complex usually is translocated to the peroxisome membrane, and a protein that is a hexamer or a dimer goes into the peroxisome as is and doesn't have to unfold into a single unit. So, and they also, as we spoke about before, some enzymes have dual localizations in the mitochondria, cytosol, and peroxisome. And then parts of these pathways are located in different organelles as well. So this is just a diagram of the peroxisome assembly, which refers to the import of these matrix proteins or enzymes, as well as the formation of the organelle. So peroxisomal proteins are numbered 1 through 26, and this corresponds to the genes that encode them, which are called PEX genes. And this model for peroxisome assembly is put together from work of many investigators working in yeast systems as well as mammalian cell culture. And if we start up here in the side, missing on the pointer. So we start here in the cytosol. We see the proteins with the PPS1 targeting signal, the proteins with the PPS2 targeting signal, and they're recognized by their cognate receptors in the cytosol, PEX5 and PEX7. And these two receptors directly bind each other as well. And the stoichiometry of this is unknown, but this is thought to be a huge cytosolic complex of interlocking PEX5 molecules with their PTS1 proteins and PEX7 with PTS2. And this is translocated to the peroxisomal membrane, probably along the cytoskeleton, and docks at PEX 13 and 14, and then this importamer complex, which is what it's called, is sort of moved over to these PEX 2, 10, and 12 proteins, and this forms a dynamic core. And then PEX5 actually is inserted into the membrane, releases its matrix proteins or enzymes inside, and then this complex out here, PEX1, 6, and 26, actually pulls PEX5 back out of the membrane and recycles it along with PEX7 for another round. So this is part of peroxisome assembly. This is the import matrix proteins. And then there are additional PEX proteins, 3, 19, and 16, that are involved in the formation of new peroxisomal membranes. So I'm just going to go through a few biochemical pathways because I certainly don't want to alienate anybody. But I think some of this is important to get a grasp on for our discussion on therapy. And so this is peroxisomal beta oxidation, and it's different from mitochondrial beta oxidation because the first step is an oxidase, as I spoke about before, which releases hydrogen peroxide, and in the mitochondria this is a dehydrogenase. And then the second two steps are similar. There's a hydratase, a dehydrogenase, and a thiolase. This enzyme, this is a single enzyme. It's called D bifunctional protein with these two functions. And what this shows us here is that the substrates for beta oxidation will enter the peroxisome through ABC half transporters that either homo or heterodimerize to form forward transporters, and probably their partner in dimerization is what dictates what substrates come in. And what I show here is the ALDP protein, which is a member of this class, or the adrenoleukodystrophy protein, and this is the protein that's defective in X-linked adrenoleukodystrophy. So this is a membrane transporter. And the substrates are saturated very long-chain fatty acids, bile acids, prostaglandins, leukotrienes, xenobiotics, dicarboxylic acids, and polyunsaturated fatty acids. And there are two oxidases, one for straight-chain fatty acids, one for branched, and there are two bifunctional proteins D bifunctional protein takes care of all of these except it's thought that dicarboxylic acids might utilize an L bifunctional protein. And this protein here, which is one of the proteins imported by PEC7, or one of the 3 PTS2 proteins, is completely redundant. And so when you have defects in the imported PTS2 proteins, and this thiolase doesn't get in, there's another thiolase that comes in on the PTS1 pathway that compensates completely. So this is a thiolase is a redundant enzyme. And then at the stage of about C16, this compound is transferred over to the mitochondria for further oxidation. So there are auxiliary enzymes involved in beta oxidation, and the point here is that there are methyl-branched fatty acids like phytanic acid that can't be beta oxidized but has to remove the methyl group with an alpha oxidation step. This is important because this is done by the enzyme phytanyl-CoA hydroxylase, also a PTS2 protein, and defects in this enzyme cause adult Refson's disease. And then you also have an enzyme called a racemase, and this is enzyme has this 2-methyl-CoA racemase has gotten a lot of literature lately because it seems to be there's an increase in gene expression in cancer, especially in prostate cancer. And this is required because both the branched chain phytanic acid and some of the cholesterol derivatives have to be racemized from their 2R to 2S isomer. And then once they get to that part, the rest of the pathway is the same. So this is the synthesis of docosahexaenoic acid, and I'm just showing you this because it brings up the point that the catabolism of very long-chain fatty acids is actually part of the process of synthesis of some of the important end products. So docosahexaenoic acid is actually synthesized in the peroxisome by virtue of a fatty acid elongation system, or synthesis of very long-chain fatty acids, which goes on in the endoplasmic reticulum. And then once you get to C24, you're actually then beta oxidized back to C22, which is DHA. So this is a catabolic step in the peroxisome that ends up synthesizing a polyunsaturated fatty acid. And there are probably many more examples of this. So this is another synthetic reaction, is ether phospholipids, and this is a very important compound. About 25% of our membrane phospholipids are ether phospholipids. We have no idea what their function is, and yet when there's a deficiency of ether phospholipids, there's a severe phenotype that you get in these peroxisome assembly disorders. So they must have an important function, and it will be very interesting to figure that out. But what makes a plasmalogen lipid is that here's your typical membrane phospholipid with a fatty acid in an ester bond at C2 and your phosphodiethanol, and you're pulling at C3 on your substituted glycerol. And in the C1 position, instead of a fatty acid and an ester bond, what you actually have is a fatty alcohol in an ether bond. And that makes an ether phospholipid, and then if you add another double bond here, you get a vinyl ether linked phospholipid, and that's what a plasmalogen is. And most ether phospholipids that are synthesized end up being plasmalogen lipids. So they're particularly abundant in myelin and RBC membranes, and when you send your diagnostic test for plasmalogen levels in your Zellweger patients or your RCDP patients, what you're actually getting is plasmalogen levels in RBC membranes. And the ideas, and there's some data for each of these, are that they function as an antioxidant by virtue of this double bond. They can function as a storage depot for docosahexaenoic acid, because when you isolate plasmalogens and you look at their fatty acid content, it seems that they preferentially store docosahexaenoic acid in this C2 position. They may also serve as lipid messengers, and an example of that is platelet activating factor, which is an etherphospholipid. And an interesting idea is that they may be involved in vesicle formation in membranes because of the steric hindrance of this molecule at the C1 position. So if you have a lot of plasmalogens that gather in the membrane, it may be that that part of the membrane is then broken for fission or fusion reactions. So peroxisome biogenesis disorders then are defects in these PEX genes that result in defective assembly of the organelle, and that's going to result in a reduced peroxisome number and reduction in matrix protein import. And the consequences are going to be multiple enzyme deficiencies, developmental defects, and progressive metabolic disturbances. And there's two groups, Zellweger syndrome spectrum and RCDP. Zellweger syndrome spectrum is probably twice as common as RCDP. Both are relatively rare for our genetic diseases. So Zellweger spectrum is a clinical continuum, and I think most of you know this. So here on the left are severe cases or classic cases of Zellweger syndrome, and they have specific dysmorphic phases, and we talked about these being the paradigm for metabolic malformation syndrome. And then sort of in the middle group here, you have an intermediate phenotype that's broadly called neonatal adrenal leukodystrophy. And I have this photo up here because something that Hugo Moser had taught me was that patients who look like they have Down syndrome but really do not have Down syndrome may have intermediate PVD phenotype or neonatal adrenal leukodystrophy. And then these two girls here clearly have the milder phenotype, which is the category of infantile Rexon's disease. And so the facial features of this morphology syndrome are clearly more recognizable in more classic cases. And the classic phenotype in Zellweger syndrome includes this neuronal migration defect. And so in column A is normal neuronal migration from levels 1 down to 6 here. And B and C are deficient migration of neurons, so it's sort of cut off here, and this is sort of abnormal migration here. And so what you end up is with polymicrogyria, pachygyria, and sometimes heterotopias. And in Zellweger syndrome, you have classically as an autopsy finding and sometimes as an MRI finding, you see these areas of polymicrogyria in the silky condition, something that's characteristic. So this just shows sort of the clinical features of this clinical spectrum that gets milder as you move out, and of which these names sort of broadly describe a severe, an intermediate, and a mild phenotype. You went over craniofacial dysmorphism, neuronal migration defects, leukodystrophy can develop at any time. Some patients in the infantile Rexon category have only cerebral atrophy. Almost all patients are retarded. There are some examples of children that aren't. They're always hypotonic and with decreased reflexes. They can develop seizures. They have a variety of different eye abnormalities. The infantile Rexon's disease cases, of course, come to clinical attention mainly for retinal dystrophy and hearing loss. There's progressive hepatomegaly that involves cholestasis and cirrhosis, and that's probably because of the bile acid defects. And there's also malformations in the development of the kidney and adrenal insufficiency, epiphyseal calcifications, and of course Zellweger syndrome is lethal in infancy, and the other disorders have variable survival. So this is rhizomelic chondrodysplasia punctata, the other peroxisome biogenesis disorder, for the other group. And this is a more homogeneous group, I think, and many more patients have a classical phenotype, which consists of rhizomelia, or shortening of the proximal long bone, so the humerus and the femur, and achondrodysplasia punctata, which is, I'll show you on the next slide. And they also have bilateral cataracts, and they have a severe growth and developmental delay. And these children here, this is a picture from the Netherlands, from the RCDP family group in the Netherlands that was sent to me. And this shows you a range of children, evidently from 2 to 13, without much change in the length of the children. And nevertheless, there are some children with a milder phenotype, and this shows one little girl who has made many more developmental milestones than most patients with possible RCDP. So this is the classic skeletal findings, and so you can see rhizomelia in the humerus, and what you also see is this abnormality in the metaphysis, so the severe splaying. And you can see punctate calcifications in the epiphyses, which looks like they're in the joints, but they're in the epiphyseal growth plate. And they're not only in the shoulder, but they're in the elbow, and they're in the costochondral junction here. And they're in the hips over here. And then what's also interesting is that you have this characteristic, what they call coronal tufting, or this longitudinal radiolucent band down the vertebral bodies. And we think that this represents a delay in ossification, and that the epiphyseal calcifications represent premature ossification. So both of those processes must be somehow going on and related to the same underlying defect, biochemical defect. And what's also interesting, I'm super sorry. I'm glad I'm not in the car. So, what's also interesting is that there's premature ossification of the intervertebral discs here, and we see this actually more often than the literature would expect us to in these children. So the RCDP clinical description then is this dysmorphic phases with frontal vaulting, with short sagging nose, anteverted nares, congenital cataracts, profound growth and developmental delays, reduced survival. They have respiratory complications, and usually the cause of death is some sort of respiratory event. And an increased incidence of cardiac and renal malformations. About 80% of them develop seizures. And neuroimaging studies, in contrast to Zellweger spectrum, have shown sort of a variety of different findings, many children having no findings at all. So when we looked for mutations in patients with peroxisome assembly diseases in these various genes, what we had to do first is take the patient's cell line, complement it with a known group, and then we would know which gene defect the patient had, and then we would look for mutations in that gene. And when we did this for more than 250 patients right now, we find that there are some groups that are much more frequent than others, and that none of the genes predict which phenotype that you have. And as a matter of fact, there is new literature that will be coming out soon, in which we have now found more patients with defects in 16, 19, and 3, which are the ones involved in membrane synthesis, and they have milder phenotypes. So in this group, we could put Z, N, and I. And probably as we get more patients, we'll have patients with infantile rexin in this group, and we certainly have all patients in this group. So the patient in the Zellweger spectrum, you can diagnose them, you have your clinical suspicion, you diagnose them based on biochemical reason, and there's no clue as to which gene is defective, underlying gene is defective. So that's a challenge to a molecular diagnosis. And that's with the exception of PEX7, in which 100% of cases of RCDP1 are caused by PEX7. And there are 2 other types of RCDP, types 2 and 3 that you've probably heard, and those are single enzyme defects in plasmalogen biosynthesis. And PEX7 is called, it encodes the PTS2 receptor. So this is the only PEX gene in which that has a consistent phenotype. So when we did this mutation analysis, just going back to this diagram of the proteins, we found that 2 mutations in PEX7 account for 65% of all RCDP cases, and this is a premature nonsense mutation and a splice mutation that encodes a null protein. And we also found, and actually this L292X mutation is common due to a founder effect in the Northern European population. And there's 2 mutations in PEX1 that account for 56% of Zellweger syndrome. And this is a missense allele, G843D, that we will revisit, and then a null allele, isoleucine 700 frameshift. And what we've also found, which I'll show you, is that association of severe mutations with severe disease. So this is probably why most patients with RCDP have classic, and the phenotype seems more homogeneous, is because the common mutation here is null. And this is probably why we're seeing many children in the milder end of the spectrum of Zellweger, because they'll carry 30% of them carry this missense allele, which in a single copy alone predicts a phenotype milder than Zellweger syndrome. So that might be the reason for the clinical phenotypes that we're seeing, or the majority of phenotypes that we're seeing. And then just to point out that this PEX1 is part of the recycling complex that pulls PEX5 under the membrane. So, in addition to the predicted consequence of the gene mutation correlating with the severity of the disease rather than the gene itself, the biochemical severity also correlates with the clinical severity. So it's likely that patients with milder disease have some residual protein. And so patients with Zellweger syndrome have the highest accumulation of very long-chain fatty acids, glycemic acids, and the greatest deficiency of plasmalogens. And the neonatal adrenoleukodystrophy phenotype is intermediate biochemically, and the infantile restless disease phenotype is the mildest biochemically. In RCDP, we see the same picture, and the milder cases have less of a deficiency of plasmalogens. And actually, there have been a few cases now of adult restless disease reported with PEX7, with an underlying PEX7 deficiency, and their plasmalogens are almost normal. So in addition to the correlation with the biochemical severity, there's also a correlation with import at the cellular level. So the phenotype correlates to the severity of the import defect, the peroxisome number, and size. And what you have here in this top panel, I'm not sure if we need to shut down the light to see this, but these are fibroblasts from control individual, severe Zellweger syndrome, and a milder infantile restless disease case. I think that's better. These, in the top panel, we're staining for the peroxisomal membrane protein, so we can get an idea of the peroxisome number and size. And in the bottom panel, we're staining for a protein that is imported in the peroxisome, so we can get an idea of how well these peroxisomes are importing. So you can see in the controls, we have this numerous 400 to 500 small peroxisomes that are all importing. And in severe Zellweger syndrome, we have much fewer peroxisomes. They're bigger, and a lot less of them are importing matrix proteins. And in infantile restless disease, we see many more peroxisomes, although they're still abnormally big, and they're not as many as normal, and many more of these peroxisomes are importing. So the correlation goes through from the molecular level to the biochemical level and to the cellular phenotype and to the patient phenotype. So just going back to some diagnostic biochemical tests, so you have a clinical suspicion of a child with a peroxisome assembly disorder, and usually you look for a characteristic biochemical profile. So you do some metabolite assays. So you look for elevated plasma very long-chain fatty acids. But since it's a peroxisome assembly disease, and you want to make sure you're not confusing it with X-linked adrenoleukodystrophy or a single enzyme defect of an oxidase or a D-bifunctional protein defect. So then you want to get you want to prove that there are a number of different pathways that are defective. And so you look at phitanic acid, and you should see that being elevated. You look at RBC plasmalogen content, and you can look at piperacolic acid. and you can do a bile acid profile. But in general, the tests most often done are plasma very long chain fatty acids, vitanic acid, RBC plasmalogens. And then these need to be confirmed with enzyme assays, and we can do these enzyme assays in cultured skin fibroblasts, choriotic villi, or amniocytes, and we usually do beta oxidation of very long chain fatty acids, that alpha oxidation of vitanic acid that I had talked about before, and the synthesis of plasmalogens. And alternatively, we could go straight to, or offer in addition to biochemical tests, the identification of 2 mutant PEX alleles. But as I mentioned before, that has its challenges itself, because the patient in the Zellweger spectrum will not tell us which gene is defective. So the approaches that have been utilized are sort of hierarchical approaches, where you look for the most common mutation first, and then you go down the pathway stepwise. So some of single enzyme defects mimic PDD, so this is why it is important to make a diagnosis of a peroxisome assembly disease based on multiple enzyme dysfunction. So defects in very long chain fatty acid oxidation, like the oxidase or D-bifunctional protein, can result in a child that looks like they have Zellweger spectrum, and defects in the single enzymes involved in plasmalogen biosynthesis in the peroxisome can result in a patient that looks like they have RCBP. So, and the other thing that's important about this is that it gives you some suspicion as to what biochemical pathway might be most important in causing the pathology of these disorders. So if the single enzyme deficiency of fatty acid oxidation can mimic Zellweger spectrum, then maybe that's the pathway that causes the major pathology. And the same thing for RCDP. And as a matter of fact, that is what we believe at this point in time, that in RCDP it's the plasmalogen deficiency that's responsible for the major pathology, and in Zellweger spectrum it's the very long chain fatty acid oxidation defect, except that this, as I showed you before, is very complicated. Because there's a lot going on in very long chain fatty acid oxidation. So the disease course is about half of the PDD patients in the Zellweger spectrum have the NALD-IRD phenotypes. So half of the patients are milder, and they do show a progressive deterioration over time. So they lose their vision, they become deaf, and they lose cognitive abilities. And that deterioration may coincide with the onset or the progression of leukodystrophy. So if efficacious treatment were available, it might halt disease progression. So when you try to think about therapies for these kids, we think in sort of two modes. One is to develop therapies that might be targeted to the metabolic defects that I just spoke about. So this is just a very simple pathway in which A goes to B, and if you have a defect, then you have an accumulation of A and a decrease in B. And so if you bring this over to the pathways that we just went through, what we could do for these kids is we could restrict their intake of dietary vitanic acid. We could reduce their intake of very long-chain fatty acids, and we could also reduce their synthesis of very long-chain fatty acids, and that's actually done with the Lorenzo's oil therapy in X-linked adrenal leukodystrophy. We could enhance omega oxidation of very long-chain fatty acids, which is oxidation from the other end instead of the end of beta oxidation. And so we could get the ER or microsomal fraction involved in the omega oxidation. Or we could supplement with product that's deficient, which might be the docosahexaenoic acid, bile acids, or plasmalogens. And a variety of these measures have all been tried, but again, it's like most of our metabolic diseases, nothing's been done in any sort of controlled clinical trial, and part of that is the problem that some diseases are so rare and the patients are spread across the world. Right now there's been 3 liver transplants for infantile Refson's disease, and only 1 reported in the literature. So we'll see what happens with that. On the one that's reported, there's not enough information to tell whether there's a change in the phenotype over time, because there's not enough time passed. And in the other case that's not reported, the child actually died of leukodystrophy after the liver transplant. So, the other idea would be to develop therapies targeted to the molecular defects. And In this area, we could induce peroxisome proliferation. And this has been done on cultured cells from patients using 4-phenylbutyrate, which is ends up being a non-specific peroxisome proliferator in this case. And we can triple the number of peroxisomes even in wild-type cells with 4-phenylbutyrate. And there's less peroxisomes in these patient cells, but they're tripled. So you just get more of a residual protein function, and so you end up with more absolute function. And so that's a possibility. And we know in cell culture that if we increase expression of partner proteins, so the PEX 2, 10, and 12 are all partners. If you get certain PEX 12 defects and you overexpress PEX 10, you can rescue that PEX 12 cell line. And the same thing with PEX 1 and PEX 6. So if we could increase expression of some partner proteins, maybe that might be a rescue. Or we could increase expression of PEX 5 receptor as a bypass. So even if the peroxisomes are not functioning well, there's some peroxisome there. And if you just keep putting PEX 5 into the system, you could get some rounds of import. And this has been shown again at the cell level by overexpressing PEX 5 in cell lines. And you could also enhance the activity of the misfolded PEX protein. And this is especially interesting in the case of the common PEX 1 G843 missense allele, and I'll talk about that in a moment. And then you could, so there might be some way to bypass the defect in a PEX protein. I guess one might be increasing PEX 5 receptor, and maybe there's some other ideas along these lines as well. So these are just some ideas. I'm sure that we can all think of more, but this would be sort of some way to approach this. And so here is a cell line of child with infantile restless disease, and it's homozygous for this PEX 1 missense allele. And what you see is at 37 degrees, here is a stain for the peroxisomal membrane, so we have a certain number of peroxisomes, we have a certain amount of import, and we have of PTS1 and PTS2 proteins, and we have no import of catalase, which is actually one of the most sensitive markers of peroxisome deficiency, and has a non-canonical PTS1 signal. And if you drop the temperature to 30 degrees, you see more peroxisomes that import more, and you can actually recover catalase import. If we turn the lights off, which it's okay, you don't need to do, you would see more catalase import here. So 30 degrees, growth at 30 degrees can rescue this missense allele, or can rescue downstream function in this cell line. And the idea is that the lower temperature is allowing more of the protein to assume a better conformation and to rescue some downstream function. And so we thought utilizing that model, and that not only has been shown for the PEX 1 missense allele, but for a number of other missense alleles in which the patient usually has IRD or at least NALD. If you drop the temperature to 30 degrees, you will get an improvement of downstream peroxisome functions. So what we did is we took a cell line from a patient who is homozygous for PEX 1 G843D, and we immortalized it, and we expressed a GFP PTS1 reporter. And the untreated cell line at 37 degrees shows no import of this PTS1 reporter, and it's cytosolic in these cells. But if you drop the temperature to 30 degrees, you can clearly see redistribution of the cytosolic reporter into the peroxisomes. And if you use non-specific chemical chaperones, which include polyols like glycerol and sorbitol, or trimethylamines like TMAO or betaine, you can also recover import of the PTS1 reporter into the peroxisome. So, so we thought that this might be a useful assay to look for if temperature can do it, and if non-specific chaperones can do it. Maybe we could use small molecules library screening and identify some compounds that might be able to do the same thing, and then could be further developed into drugs for clinical use. So the next thing we had to do, though, is to make sure that that redistribution of reporter from the cytosol to the peroxisome did indeed coincide with improvement in peroxisomal functions, even though there was improvement in import. So here just shows that we did indirect immunofluorescence on primary cells containing at least one G843D allele, and showed that with these nonspecific chaperones we could recover catalase and biolase import from baseline. And we also did enzyme assays, including plasmalogen biosynthesis. And in this assay, the normal number that you come up with is less than 0.7. And in the untreated cells, it's abnormal, up at 2.26. And with 30 degrees and with these nonspecific chaperones, we can then decrease that. So there's an improvement in plasmalogen synthesis as well. So we also showed that by immunoblotting that you can recover biolase processing. So that's a PPS2 protein. It gets into the peroxisome, and the leader sequence is cleaved, and so you have a change in size from a 44 kilodalton protein to a 42 kilodalton protein. And so this, we thought that this assay should also reflect the increase in biolase import that we were seeing in the IF and the improvement in plasmalogen synthesis. And sure enough, we, again in primary cell lines, this is the wild type, which is the 42 kilodalton. And then this is the 30 degrees, which is also this like Western blot thing, but this is the same size as this. So at 30 degrees, we have all of the protein in the 42 in the processed form. And in untreated, all of the protein is in the unprocessed form. And then with various treatments, you can see that there's partial processing of biolase. It's just actin. And this is a cell line with one G843D allele, and we can see that this cell line responds to, but probably not as well as, two G843D alleles, which makes sense. So we figured that this assay was ready for prime time because the change in phenotype was correlated to change in function. And so we set up a high throughput phenotype assay in 96 well plates. And the cells were incubated in chemical for 2 days, and we used duplicate plates. And for our each plate had a negative and a positive control. And for our negative controls, we have no treatment, which is the DMEM media, and the treatment with the solvent for the chemicals, which is 0.3% DMSO. And for our positive controls, we had those nonspecific chemical chaperones that I just showed you, the TMAO and the glycerol. And we screened 2 libraries. This is a very small screen, a library of 2,000 compounds of known bioactive compounds, and a small kinase inhibitor library. And we grew up the cells in chemical for 2 days, and then we fixed them and we stained them with a nuclear stain, and we imaged them on a BD Biosciences imager. And this was actually done at Hopkins and was completed just over the past year. And we took the imager took 3x3 montage images in both the GFP and the DAPI channels. And then we evaluated the data by visual inspection and compared them to the positive and negative controls on the plate. And it would be nice, of course, to have developed some software in which we could have the computer figure out whether it's recovered or not. So these are images from the phenotype screen, and you can see the untreated cells and the positive controls with a lot more punctate cells here. And then this is a well that we would score as negative, and this was a well that we scored as positive. Okay. So, and then these were the compounds that came out of this screen as being positive and that we're working with further. And again, this is your negative control, this is your positive control. This is a 10 micromolar acetate diacetate. This is a flavonoid compound in the flavone class. And this is 10 micromolar epicholestanol, which is actually it looks closest to a bile acid. That's the type of sterile structure it has. And these are 2 kinase inhibitors that were both PKC inhibitors, and so we're calling them PKC inhibitor 1 and PKC inhibitor 2. And we have done some confirmation. We haven't completed the confirmation of these compounds, but this is an example of going back to the primary cell lines, growing them in these compounds, and looking at plasmalogen biosynthesis. And we did see in 4 different replicate experiments and averages, we have a significant value on our student t-test for improvement in plasmalogen synthesis in these 4 compounds. And what's really interesting about this is that PEX1 and PEX6, I didn't mention earlier, are AAA ATPases. And so they heteroallegorize, bind ATP, and hydrolyze ATP, and that energy of hydrolysis is translated into motion of the molecule, and that's how it pulls PEX5 out of the membrane. So it's an ATP binding protein, and it turns out when you read through the literature on these flavones and kinase inhibitor, is that both of those molecules have the property of binding at ATP binding sites. So our working hypothesis at the moment is that these chemicals are binding at the ATP binding site of PEX1 and helping it fold better. So the binding itself is acting as some sort of chaperone, and it must be some sort of competitive binding. So it binds, it folds better, and then there's a competition with ATP, and ATP binds, and this molecule must fall off. So that's our working hypothesis. So that brings us back to the pathogenesis of peroxisome biogenesis disorders. So we can work on therapy for these patients, but we also want to understand the disease process, and we have no idea, as I mentioned before, how these biochemical pathways are causing disease. And we also believe that when we understand this, that we'll have more targets for therapy as well. So the tools so far that are being used are investigations in patients, mouse models, and mammalian cell culture, and people are looking at a number of different things. And we've been involved also in a PEX7 deficient murine model to evaluate the role of plasmalogens in organ development. And that's it. Just let's get some of my collaborators from McGill and from Hopkins, and then of course the parents and patients and the funding sources.
So in your screen for compounds that rescued your cell lines, it looks like with the ones that you screened positive, it looks like some cells had a whole lot of staining where other cells had none. It looked like So do you have some idea about what's happening?
Right. That's really interesting. So this happens in cell lines from the patients themselves, and we call this peroxisomal mosaicism, although it's not a mosaicism at the genetic level, but it's a mosaicism in the cellular phenotype. So in the milder patients, we'll have some cells that actually recover import, or some cells that you can see importing proteins in your stain right next to cells that aren't importing proteins. And we have no idea why that's happening. And we've tried actually cloning those cells and seeing what happens to them over time. And that's been sort of conflicting. But it seems like the cells with the peroxisomes end up over time losing their ability, with the ability to import, because they all have peroxisomes, lose their ability to import over time. So I have no idea what's going on. But again, I see that, and if I put the cells at 30 degrees, over time, I see this redistribution of the GFP-PTS1, with some cells doing better than the others. Also the primary cell lines, to just look at the IF endogenously, the same thing happens. It's sort of an overtime process. So I think that's the same thing that's going on with the chemicals, is that over time, the cells are moving towards complete redistribution, which we can actually get at 10 days at 30 degrees.
So what's the thinking as far as what component of peroxisome function needs to support the features?
Well, that's the pathogenesis part. So, if you talk about, so I think there's a combination of the extreme hypotonia and some, maybe is Margaret here? You can help me with this. So this extreme hypotonia, and I think that's part of the long phase of epithelial folds, there's also a underlying skeletal defects. That might be part of the wide open anterior fontanelle that you see. And why you end up with glaucoma and cataracts, that I don't know, but over time there's degeneration. These are degenerative processes too. So there's degeneration of the retinal pigmentary changes. And the liver disease, I think, is a progressive disorder due to the bile acid synthesis. The microcysts and tubular defects in the kidney, I don't know. That's really interesting.
What part of the hearing is affected?
It's usually sensory neural hearing loss, and it's progressive in the infantile reticence patients.
I was struck by the comparison you drew to Down syndrome.
That one case.
Are any of these genes on 21?
That's a good question. I don't know the answer to that. I'll have to go back and look. I know that there's chromosome 12, there's two of them on chromosome 12, and there's one at 17, and there's one, I don't remember. I'll have to go back and look.
I think since the disease has been considered a protein misfolding problem, so all your IC done, do you think it's possible the compound also acting on the ER unfolding response possibly?
Possibly, but one of the things about most peroxisomal, most PEX proteins and peroxisomal matrix proteins are that they're not associated with the ER, so they're translated on smooth endoplasmic reticulum in the cytosol, and they're picked up by those receptors in the cytosol. So they don't go through the ER, except for those three that are involved in membrane formation. They have a route through the ER. So I don't think they're involved in the ER unfolded response because they don't go through the ER.
I'm just curious that you show one of the compounds is epicholesterol.
Yeah.
Have you think about it? Why that helps sampling? Is it possible? No, some cholesterol oxidation about cholesterol ester is happening in peroxisome. And also there are some studies like give patient with statin helps.
Mm-hmm.
Do you think there's any maybe act as a substrate somehow help the sampling?
Yeah, I'm not sure that epicholesterol is going to come through our confirmatory assays, which is why I didn't really talk about it. But also, I've looked back through the literature, and I can't actually make a working hypothesis if that was working, how it would be doing it either, because it doesn't have the properties of binding to either to any proteins that have ATP binding sites like these others.
Yeah.
There are some examples, there are some papers in the literature that talk about membrane lipids, and epicholesterol as a membrane lipid can actually influence the activity of the magnesium, calcium, ATPase in the membrane by some sort of modulation that's unknown. But that's about the closest I can get. But yeah, we can talk about that.
I just think the PKC inhibitor could have a lot of other functions.
Mm-hmm.
Don't you worry that.
That's absolutely true. So the idea that there's a pathway involving protein kinases as an alternative hypothesis in the peroxisome is very interesting as well. There's also something different. I have some other slides about the way the peroxisomes look in the primary cells after being grown in kinase inhibitors. So I think it's proliferating the peroxisomes, and I also think they're abnormal appearing.
So before we go, do we have anybody from Tulane?
No.
Okay. All right. Well, I think Nancy will be around, so if there's any more questions, feel free to try to work in. And thank you for your. Did I inspire?